Single molecule analysis within microdroplet environments is an exceptionally innovative approach that opens a window into the hidden world of individual molecular events. In traditional experiments, measurements are taken from bulk samples where millions of molecules act simultaneously, and their individual behaviors are averaged out. In contrast, by isolating single molecules within microdroplets, tiny compartments, typically in the picoliter to nanoliter range, researchers can study the unique dynamics, interactions, and reaction kinetics of individual molecules with remarkable sensitivity and clarity. In these microdroplet systems, each droplet acts as an independent, self-contained reaction chamber. Imagine each droplet as a miniature test tube where a single molecule or a very few molecules are encapsulated along with the necessary reagents. Because the volume is so small, the local concentration of any fluorescent label or reaction product is significantly enhanced, which dramatically increases the signal-to-noise ratio. For example, consider a protein that undergoes a conformational change upon binding a ligand. By confining a single protein molecule within a droplet and tagging it with a fluorescent probe, any change in the fluorescent signal due to folding or binding can be monitored in real time with high precision. This level of detail is similar to watching a single dancer perform on a spotlighted stage rather than observing a large, indistinct crowd where individual movements are lost. One might ask, why is it so crucial to study molecules individually? The answer lies in the heterogeneity inherent in biological systems. Even genetically identical molecules can behave differently due to local environmental fluctuations or random stochastic events. Bulk measurements average these variations out, masking rare but significant events that could be critical for understanding molecular mechanisms. For instance, in enzyme kinetics, a small subset of enzyme molecules might follow an alternative reaction pathway that leads to different catalytic efficiencies. Single molecule analysis in microdroplets can capture these rare events, providing insights into reaction intermediates, transition states, and even transient conformations that are essential for elucidating the full picture of molecular function. The creation of microdroplet environments is typically achieved using advanced microfluidic devices. These devices feature intricately etched channels and junctions that allow immiscible fluids, often an aqueous phase containing the molecules of interest and an oil phase, to be precisely combined. At a flow-focusing junction, the aqueous solution is pinched off by the continuous oil phase, forming a series of uniformly sized droplets. This process is highly controlled. By adjusting flow rates, channel geometries, and surfactant concentrations, researchers can produce droplets that are nearly identical in size and composition. Imagine an assembly line where every container is filled with exactly the same amount of liquid. This uniformity is critical for ensuring that each droplet provides identical reaction conditions, thus allowing direct comparisons between individual molecular events. Within these droplets, several techniques are used to monitor single molecule events. Fluorescence microscopy, particularly total internal reflection fluorescence, TIRF, microscopy, is one of the most common methods. TIRF microscopy excels at reducing background fluorescence by only exciting molecules near the surface of a droplet, making it possible to detect the faint signals emitted by single molecules. For instance, in studies of DNA-protein interactions, a fluorescently labeled DNA strand can be confined in a droplet, and its interaction with a binding protein can be observed as changes in fluorescence intensity or wavelength. Another technique, single molecule Forster resonance energy transfer, SMEF-FRET, relies on measuring the energy transfer between two fluorescent labels placed at different sites on a molecule. As the molecule changes conformation, the distance between these labels changes, altering the FRET efficiency. Observing such changes within a microdroplet environment enables researchers to capture real-time conformational dynamics with unprecedented resolution. A common question that arises is, how do scientists ensure that each droplet contains only a single molecule? This is achieved by a method known as limiting dilution. By diluting the sample to very low concentrations before encapsulation, the probability of having more than one molecule per droplet becomes very low, according to Poisson's statistics. For example, if a solution is diluted such that the average number of molecules per droplet is 0.1, then most droplets will contain 0 molecules, and a small controllable fraction will contain exactly 1 molecule. This statistical control is essential for ensuring that the measurements reflect single molecule behavior and are not convoluted by multiple interactions occurring simultaneously. In addition to optical methods, other sophisticated techniques such as optical trapping and nanopore sensing are also integrated into microdroplet platforms for single molecule analysis. Optical trapping uses highly focused laser beams to hold and manipulate individual molecules or nanoparticles in place. This allows researchers to apply mechanical forces to single molecules, such as stretching a DNA strand or unfolding a protein, and directly measure the forces involved in these processes. For example, by trapping a single polymer molecule within a droplet and gradually increasing the laser power, scientists can determine the precise force required to induce a conformational change. Nanopore sensing, on the other hand, involves passing individual molecules through a nanoscale pore and recording changes in ionic current. Each molecule produces a distinct blockade pattern as it translocates through the pore, allowing for the determination of its size, charge, and even structural features. When combined with microdroplet techniques, nanopore sensors can analyze molecules in a highly controlled and isolated environment, minimizing interference and enhancing resolution. Another fascinating aspect of single molecule analysis in microdroplets is the ability to conduct time-resolved studies. Because reactions are confined to the small volume of a droplet, it is possible to initiate a reaction at a well-defined moment, say by merging two droplets containing different reactants, and then observe the resulting molecular dynamics in real time. High-speed cameras and sensitive detectors capture these events as they unfold, providing kinetic data that reveal the rates of binding, unbinding, conformational changes, or chemical transformations at the single molecule level. For instance, in the study of RNA folding, researchers might trigger folding by rapidly mixing a denatured RNA solution with a refolding buffer in a microdroplet. The subsequent changes in fluorescence can then be recorded at millisecond intervals, allowing scientists to map out the folding pathway and identify intermediate states. An important question to consider is, what are the benefits of analyzing molecules one at a time compared to bulk analysis? In bulk experiments, the behavior of millions of molecules is averaged, which can obscure the presence of subpopulations with unique behaviors. Single molecule techniques, however, reveal the full distribution of molecular responses, capturing rare events such as transient intermediate states or variations in reaction rates that could be critical for understanding diseases or developing new drugs. For example, in drug discovery, identifying a rare conformational state of a protein that is the target of a new therapeutic agent might be crucial for designing more effective inhibitors. Single molecule analysis provides that level of detail, potentially leading to breakthroughs that would be impossible to achieve with bulk methods. The applications of single molecule analysis within microdroplet environments extend across multiple fields. In biophysics, studying individual protein folding events can reveal the mechanisms by which misfolding leads to diseases like Alzheimer's or Parkinson's. In enzymology, measuring the catalytic activity of a single enzyme molecule can provide insights into how enzymes achieve such high efficiencies and how mutations might affect their function. In nanotechnology, the precise control and analysis of single nanoparticles can lead to the development of novel materials with custom-designed properties. Moreover, in clinical diagnostics, single molecule detection methods are being developed for early disease diagnosis by detecting minute quantities of biomarkers, such as circulating tumor DNA or single viral particles, which can be critical for early intervention and treatment. A thought-provoking question is, how do researchers deal with the enormous amounts of data generated by single molecule experiments in microdroplets? The answer lies in advanced data analysis algorithms and machine learning techniques. High-throughput microdroplet experiments can generate terabytes of data, as each droplet's time series fluorescence or current trace must be analyzed to identify individual molecular events. Researchers employ automated data processing pipelines that use statistical methods and machine learning to sift through the noise and extract meaningful signals. These computational tools not only speed up the analysis, but also help in identifying subtle patterns and correlations that might be overlooked by manual inspection. Furthermore, the integration of microdroplet-based single-molecule analysis with lab-on-a-chip technologies is paving the way for new diagnostic platforms that are both sensitive and portable. For example, microdroplet systems can be used to perform digital PCR, polymoleic rack chain reaction, at the single molecule level, enabling the detection of rare genetic mutations in a patient's blood sample. This could revolutionize personalized medicine by providing rapid, accurate diagnostics that guide targeted therapies. Imagine a handheld device that can perform thousands of individual molecular tests simultaneously, each in its own microdroplet, and deliver results in real time. This is the promise of single molecule analysis in microdroplet environments. There are also exciting developments in combining single molecule techniques with other modalities of analysis. For instance, researchers are exploring ways to integrate mass spectrometry with microdroplet platforms to identify the chemical composition of individual molecules. In such systems, droplets containing single molecules can be directed into a mass spectrometer where the molecules are ionized and their mass-to-charge ratios are measured. This integration offers a powerful means of characterizing molecules with both high sensitivity and specificity, and it could lead to new insights in proteomics and metabolomics. Despite these impressive advances, several challenges remain in the field. One of the primary difficulties is maintaining the stability of microdroplets over extended periods, especially when experiments require long observation times or involve multiple sequential reactions. Droplet coalescence, evaporation, and unwanted interactions with the channel surfaces can all affect the reliability of the measurements. Researchers are addressing these issues by developing improved surfactant formulations and microchannel coatings that reduce droplet instability. Additionally, the design of the microfluidic chip itself is continuously evolving to incorporate features that minimize these issues, such as integrated valves, flow-focusing junctions, and temperature control elements. Another challenge is the precise control of molecule encapsulation to ensure that each droplet contains exactly one molecule. While dilution and Poisson statistics are effective, achieving absolute single-molecule occupancy in every droplet remains a technical hurdle that researchers are actively working to overcome through innovative chip designs and sample preparation techniques. For instance, recent developments in droplet sorting technologies allow for the selection and isolation of droplets that contain a single fluorescent event, thereby refining the dataset for single-molecule analysis. Looking ahead, the future of single-molecule analysis in microdroplet environments is extremely promising. With ongoing advancements in microfabrication, optical detection, and computational data analysis, it is likely that we will see even more sensitive and high-throughput systems that push the limits of what is detectable. Innovations such as droplet microarrays, where thousands of droplets are mobilized on a chip for simultaneous analysis, and fully integrated lab-on-a-chip systems that combine sample preparation, reaction initiation, and detection on a single device are on the horizon. These technologies will not only accelerate basic research in molecular biology and chemistry, but also lead to practical applications in clinical diagnostics, drug discovery, and environmental monitoring. In summary, single-molecule analysis within microdroplet environments is a transformative technique that offers unparalleled insights into the behavior of individual molecules. By confining reactions to tiny, uniform droplets, researchers can significantly reduce background noise, achieve high detection sensitivity, and capture the dynamic processes of molecules in real time. This approach opens up new possibilities for understanding complex biological mechanisms, developing advanced diagnostics, and creating innovative materials and therapeutics. The integration of cutting-edge optical methods, precise microfluidic control, and sophisticated data analysis tools makes this field a vibrant area of scientific exploration with far-reaching applications.